Health and medicine. Whenever we get sick, we consult a doctor. The doctor gives us medicines, and we get well. How has this thing made possible? The answer to this question lies in biology. It is only because of biology that the study of various disease-causing microorganisms has been made possible. Researchers have investigated the biology of microorganisms and have discovered the methods of controlling the diseases. Even the formulation of drugs for fighting the disease-causing microorganisms has been made possible because of the study of biology of those microorganisms. Pharmacognosy is a name derived from Greek words, which mean drug and knowledge. In the field of science, researcher deals with the secondary metabolites found in many plants, animals, and microbial natural sources. The American Society of Pharmacognosy, ASP, defines it as the study of physical, chemical, biochemical, and biological properties of drugs, drug substances, or potential drugs, or drug substances of natural origins, as well as the research for new drugs from different natural sources. Most of the countries in the Southeast Asia region have a heritage of traditional medicine system. According to the recent WHO report, about 80% of the world population is still using natural product for their primary healthcare needs. Pharmacognosy can provide safe and effective drugs in combination with modern medical systems. Like the first medicines, many of today's medicines are made from plants. The chief ingredient of aspirin, salicylic acid, was originally obtained from the bark of willow tree. The willow is the genus Salix, hence the same name salicylic acid. The bark of the cinchona tree, Cinchona sp., contains quinine, a drug used to treat malaria. Digitalis, which is used in treatment of heart disease, is obtained from the leaves of foxglove plant, Digitalis purpurea. Periwinkle, Catharanthus sp., is the source of two drugs that are effective against Hodgkin disease and some leukemia. Two powerful pain relief compounds, morphine and codeine, are extracted from opium poppy, Papaver somniferum. Medicinal animals are important resources linking people to environment, and their use promotes the traditional knowledge related to them. Over 50% of commercially available drugs are based on bioactive compounds extracted from the non-human species. Almost every class of drugs includes a model structure derived from nature, exhibiting the classical effects of specific pharmacological category. A great number of these natural products has come to human society from the scientific study of remedies traditionally employed by various cultures. In addition to plants and microbes, there has been increasing attention paid to animals, both vertebrates and invertebrates, as sources for new medicines. The healing of human minor illness by using therapies based on medicines obtained from animals or ultimately derived from them is known as zootherapy. Animals have been methodically tested by pharmaceutical companies as sources of drugs from modern medical science, and current percentage of animal sources for producing essential medicines is quite significant. Some examples of animal-based medicines are leeches, viper venom, maggots, spider silk, salmon, and horseshoe crab. Leeches. Leeches are slimy bloodsuckers that can eat 10 times its own body weight of blood. Today, medicinal leeches are used after severe trauma to help reattach digits, close wounds, and help mend skin after plastic surgery. The leech, Hirudo medicinalis, is approved by the Food and Drug Administrations for the medical use. While the leech feeding on the surface of the skin, they also secrete anticoagulants, which help to keep the blood flowing. The blood pools in the reattached thumb can be sucked out by the leech until the veins redevelop. Viper venom. The molecule called bradykinin from the viper venom is found to be related to a class of molecules that stop angiotensin-converting enzymes, ACE, from blocking bradykinins, a protein that causes blood vessels to dilate and lower blood pressure. Bradykinins were eventually developed into the drug captopril, used to treat hypertension, cardiac conditions, and to preserve kidney functions in diabetics. Maggots. Maggots are small, voracious eaters that feed on diseased and dying flesh. Their feeding habit is a life-saving asset for those suffering from chronic wounds and infections. Maggots can turn a chronic wound into an acute wound in a matter of days by eating the chronic tissue and bacteria. From there, the wound becomes treatable and can finally heal. These hungry insect larvae are sterile, work quickly, and cost less than traditional treatments. Spider silk. Spider silk is referred to by many scientists as biosteel. Much like in Spider-Man movies, spider silk has super tensile strength. Spider silk can also be used to make artificial ligaments and tendons that support tissues, bones, and nerve cells, holding them steady while they grow. These artificial silk parts then fall apart gradually, after the cells have been given enough time to grow. The spider silk gene for the silk protein is connected to DNA in the mammary gland of goat. The cell of the mammary gland is then combined with an egg to ultimately produce an embryo that has the gene incorporated into its DNA. The silk protein is then made when the female starts lactating. Salmon. Salmon belongs on a dinner plate, but it also can be found in the pharmacy, too. They do produce calcitonin hormones to regulate their own calcium levels from an endocrine gland in their neck. Humans make calcitonin, a hormone that inhibits bone loss, in the thyroid gland. But in postmenopausal women and people with Paget's disease, the rate of bone loss increases. The synthetic version of this calcitonin from the coho salmon, the calcitonin salmon, can be used to treat people with calcium regulation disorders. Horseshoe crab. Over millions of years, the horseshoe crab has been exposed to an awful lot of microbes, making them immune to a wider range of threats than any other animal. Every person in the world today who receives vaccines, antibiotics, or implanted medical devices such as pacemakers has had their safety ensured by the blue blood of the horseshoe crab. Protein in the blood called limulus ambisite lysate, LAL, reacts to all kinds of microorganisms that can easily detect dangerous endotoxins that cause fever and can be fatal. Scientists observed that the blood reacts to any contamination.